So we left off talking about DNA viruses and then kind of stopped there. Now, today, we're going to go into some of the different DNA viruses that are particularly ones that impact humans. So they infect us. And I'll start with the enveloped DNA viruses first, and then we'll go into the non-enveloped. Pox virus is an example of an enveloped DNA virus. And it's a really large and complicated virus. It's a double-stranded DNA virus. So remember, most DNA viruses are double-strand, not all. Some can be single. Most RNA viruses are single-strand, not all. Some can be double. But there isn't a virus that has both DNA and RNA packaged into its virion. Okay. What's unique about, well, maybe not unique, but what's rare about the pox virus is that it multiplies in the cytoplasm. And per the quiz that we just had, generally speaking, RNA viruses multiply in the cytoplasm, and DNA viruses multiply in the nucleus. And so why is that happening? Well, to understand transcription, we need to talk about how RNA viruses can do that without having to be in the nucleus. RNA viruses encode their own RNA-dependent RNA polymerase. Remember, the polymerase is the enzyme that transcribes RNA off of a DNA template, right? Most DNA viruses just enter the nucleus of the host and use the host's DNA-dependent RNA polymerase to transcribe. But because the pox virus is so large, it encodes its own DNA-dependent RNA polymerase. So it doesn't need to go to the nucleus. It goes into the cytoplasm, it unpackages, and then there's an early transcription to then start making these different proteins. And one of those proteins is the polymerase, and then that starts replicating. And so it's it doesn't need to go into the nucleus, right? Smallpox was, if you remember, the first virus that had a really well-established vaccine from Jenner. And so the result is that now it's pretty much eradicated from the planet, fortunately, because it is a very nasty virus that does lead to, if it doesn't kill you, it can permanently scar you for life. In the case with this image of this poor little one here. The vaccination has pretty much been non-existent since the 70s, because by 1980, smallpox was claimed eradicated. And there is, since 2002, been a reintroduction in military personnel or medical personnel that are traveling abroad. Because while smallpox has been eradicated, it's not that it's actually no longer on the planet, right? It's still around. It's just not circulating in humans, and so as much. And so it's nice because the vaccine is kind of a painful, unpleasant experience. They have this two-pronged needle, and they put the vaccine on there, and then they puncture your skin a bunch to get like an open wound to deliver it that way. And actually last fall, I had a veteran who was a student of mine, and he said that he had to get this vaccine and that it wasn't very pleasant.
Quick question, why the prong? I mean, couldn't they make it into like a serum or something and inject it like they do every other vaccine?
Yeah, I don't know why. I mean, so if you look here where it infects, it infects, sorry guys, just one second. It infects the skin. And so I think what they do is they must decide to deliver to the skin because that's the site of infection, rather than, most vaccines, they just stick into your muscle tissue and shoot it there. I don't know why they have this pronged approach, actually, to be honest, but that's a good question. So sorry, I don't have a great answer for you there. Any other questions? Okay, feel free to interrupt at any time. Herpes viruses are another example of enveloped DNA viruses. And all of them can be latent, meaning they go into kind of a dormant state and then infection comes later on in the lifespan of the patient. And when we talk about this recurrent infection that happens later on, it usually comes about because the person is aging and they have a decreased immune system to keep that at bay, or they're experiencing chemotherapy for cancer, which is destroying their immune cells as well, or they have some other kind of immunocompromisation that then allows this virus that's been kind of laying low to flourish again. And herpes viruses are one of those opportunistic pathogens that can lead to the demise of patients with AIDS. It has a large envelope. The capsid is icosahedral, not helical, and it has double-strand DNA. This replicates within the nucleus because most, as we said, most DNA viruses replicate in the nucleus. And so it has inside here's the DNA, here's the capsid. On the outside, we have the envelope, and then in between is what's called the tegument. And that's the matrix proteins and stuff that are there in between the envelope and the capsid itself. Okay.
You said that not every virus has the matrix proteins though, right?
Correct. Not all viruses will have matrix proteins. Most envelope viruses will have some kind of matrix protein in between the envelope and the capsid, but certainly naked viruses are not going to. Good clarification. So HSV1 and 2, those are the type of herpes viruses that cause things like cold sores and genital lesions, respectively. HSV3 is going to be varicella, also known as chickenpox and shingles later on. Herpes 4 is going to be Epstein-Barr virus. And so that's going to be, think of mono when you think of Epstein-Barr virus. HSV5, that's cytomegalovirus, which I mentioned a while back, is particularly harmful to developing fetuses if they were to contract that from their mother. And then we have human herpes viruses 6 and 7. Those are responsible for roseola, which is kind of a rash that infants get, like a very speckled red rash. I don't know if it's uncomfortable to them. It doesn't seem to agitate them, but they're definitely sick. They have a fever with that as well. And then herpes virus 8, that's going to be associated with Kaposi sarcoma. So a cancer of the connective tissue, bone, ligaments. And so as you can see, all of these HSVs can be pretty varied in what type of tissues they infect. They're all within the same family, but some infect different types of epithelial cells. Some infect your lymphoid tissue, salivary glands, connective tissue. So it's a broad range of different types of tissue tropisms for these viruses in this family. As I said, HSV1 is going to be like cold sores, fever blisters. Oftentimes, children get HSV1. HSV2 is going to be the genital lesions and sometimes oral lesions, and that's obviously usually later on in life between ages of 15 to 30, because that's when people start becoming sexually active, and that's when that can spread. And it's important to know that HSV2 can spread without discernible lesions. Okay, if a person has HSV2, they may still be able to pass that on, even if there is nothing to show that they have that infection. Herpes labialis, that's the fever blisters, the cold sores right around here. Herpetic gingivostomatitis is going to be a very uncomfortable, unpleasant inflammation of the mouth. And that's in this picture here. You get these like lesions on the tongue and on your lips, and it's very uncomfortable, as is herpetic keratitis or ocular herpes, which is the herpes of the eyes, which makes it feel very gritty when you blink and can lead to conjunctivitis. So, the nasty buildup in your eyes, very painful, sensitivity to light. And then genital herpes. That's typically going to start with just feeling bad, a fever. That might lead to swelling and tenderness in the groin, and then gets more and more painful in that area when you're urinating, gets itchy. And usually the first bout with it is the worst. And over time, it can be triggered by things in females, by menstruation, or things like stress, or if you're infected with another type of microbe, like a bacterial infection, which is weakening your immune system, allowing that herpes virus to now again flourish. But usually subsequent outbreaks aren't as bad as the initial one. Herpes in newborns can be lethal. It's dangerous. And so this can happen during development, or it can happen during the birthing process. And it can infect all over them, in their mouth, their skin, their eyes, their central nervous system. And so there is some screening in pregnant women, but it's not, my understanding is not a routine screen. So if a patient comes and is in labor and there are visible lesions, then they will either treat that patient with like acyclovir, or they'll try to do a C-section to prevent the infection through the vaginal canal. But as we've mentioned, it can be spread without discernible lesions present. So, sometimes it misses the radar. Okay, varicella, think chickenpox. And maybe none of you have had chickenpox because now there is a vaccine for it. When I was a kid, there wasn't a vaccine. And so we all got chickenpox at some point, and it was itchy and painful, and you weren't allowed to scratch it because then you could get a scar. And so most people my age or older have scars, not like pox or anything, but some scars around from picking at a chicken pox. Note that it's not a pox virus though, even though it's called chicken pox, because it does have that pock-like display on the outside. It's not a pox virus, okay? It's a herpes virus. And this is one that can, once you get it, you have it. And they actually used to have chicken pox parties, I believe, back in the day, where if one kid in the neighborhood got it, then they would just get all the kids together and have a sleepover and be done with it, right? Everyone gets chickenpox. It can remain dormant and then re-establish itself as an infection later on in life in the form of shingles. So usually older patients will have a re-outbreak of varicella, not in the form of chickenpox, but in the form of shingles, which has a little bit different manifestation.
But you can't get shingles if you didn't have chickenpox, correct?
Yeah. I mean, so before they had a vaccine, it was important actually that children got chickenpox, because if they didn't, chickenpox in an adult could be a much more severe reaction. So kids are more hardy and they don't get nearly as bad of an infection. Okay, yeah. So Hava had a chickenpox party even in the late 80s. Okay, cool. So that is something that happened. So yes, if you have shingles, that means that you had chickenpox at one point. Okay. Now, if you've never been exposed to varicella and you don't have the vaccine, then you get exposed and it will manifest as chickenpox initially. And as I said, in an adult, it's going to be a lot worse, more aches and pains, fever, elevated and stuff like that. So you won't, I don't believe that it will skip over the chickenpox and just go to herpes, or I mean to shingles. Does that answer your question?
Yes, it's perfect. Thank you.
Hepatitis, right? Hepatitis is an enveloped DNA virus. And interestingly, fun fact, it's never been grown in tissue culture, which makes it difficult to study. And another fun fact, which I think is even more fun than the never being grown in tissue culture, is that it's one of these weird ones that has both double and single-stranded DNA. Okay, so again, like I say, in biology, there's always these exceptions. This isn't the DNA RNA exception, but it's a virus. It's a family of viruses that have double strand and single strand DNA packaged into their viral particle. Okay, so a little bit of an odd one. And it gets its name hepatitis because it has a tropism for the liver. So it inflames the liver, hence hepatitis. There are many different varieties, and we'll go through these briefly. Hepatitis B is going to be in humans, and that is the DNA virus. Now, it does get a little bit confusing because hepatitis A, C, E, and D are all RNA viruses. So not all hepatitis viruses are these double and single strand DNA viruses. Some can be RNA, and the reason they're called hepatitis is because of the tropism. Just like chickenpox isn't a pox virus, it's called chickenpox because of the manifestation of it. These hepatitis viruses are called hepatitis because they are infecting the liver. So hepatitis A is from food that's contaminated. That's not typically a deadly one. Hepatitis C, however, that comes from blood transfusions or IV drug use, and so that can wreak havoc on your liver, and you can get cirrhosis, and people will succumb to that if they're not careful, especially because hepatitis C often is acquired through lifestyles that are hard on the body as well. So if you're using drugs or using alcohol, and that's going to have a certain impact on your liver. And then on top of that, you now have a virus that's impacting your liver. It's a double whammy. Hepatitis E comes from food as well that's contaminated with feces. It's a relatively new one of these. And then hepatitis D is going to be in cells that are already infected with other hepatitis B viruses. Okay, so it's kind of like a satellite virus in a way, because its RNA isn't capable of setting up shop and replicating by itself. It needs the help of Hep B to produce the different enzymes it needs to replicate its genome. Any questions on that so far?
Doc, which one were you talking about last week that we just found a cure for?
Hep C. So the Nobel Prize went to researchers that worked with Hep C, and they do have a vaccine for that now. I don't know how widely implemented it is. I don't know what they've been doing with it, but there is now a vaccine for it, luckily. Thanks for bringing that up. It's a good point. I forgot to mention that. So that was spotlighted this week. Okay, so this is just some information on Hep B. It's a lot of text. I'm not going to just read all this to you. It's really for your own information. Most viruses are going to cause malaise and fever and chills and discomfort, right? So these are common symptoms, and I'm not expecting you to memorize that it has an average 7-week incubation, for instance. All this is here just for your own education. Okay. Same with this, right? You don't need to know that the vaccines were derived from antigens that were cloned in yeast and that there are 3 doses with boosters. This stuff is purely just FYI. So I won't really go through that. Now let's talk about non-enveloped DNA viruses. These are going to be adenoviruses, papilloma, polyoma, and parvo, okay? So they have DNA genomes, but they do not have envelopes. Adenoviruses have about 30 different types that are associated with pathology in humans. And remember, the adenoviruses, they infect the adenoids, or the lymphoid tissue, and they have satellite viruses that come along with them, the adeno-associated viruses that we talked about on Monday, briefly. In addition to the lymphoid tissue, they can also spread by respiratory droplets, ocular secretions, and can cause things like the common cold, pharyngitis, conjunctivitis, and acute hemorrhagic cystitis. So varied different types of symptoms and signs within these different 30 types of adenoviruses, okay? Severe cases are treated with interferon. And what's interferon? Somebody?
Oh, that's a great question.
It's a signal.
Yeah, I mean, so it was the answer to a quiz question today. But one of the things interferon does is it signals to cells nearby that a cell itself is infected. And so then that nearby cell can start transcribing and upregulating the production of innate antiviral molecules. So interferon is important for the innate immune response. Papilloma and polyoma viruses are circular DNA, okay? So it's kind of like a plasmid. Remember I said a plasmid was a bacterial genome that's circular. With these viruses, their genome is also circular, and they can lead to persistent infections and tumors. Both polyoma and papilloma can cause different types of tumors. The papilloma virus is also the wart virus, right? Over 100 different strains of HPV. And this can be transmissible through direct contact or contaminated fomites. Anybody know what a fomite is? maybe hear the word fomite in the COVID-19 era. This is a fomite, this mouse pad. Doorknobs are fomites.
Like a vortex?
Not a vortex like this right here. This is a fomite. It's a surface that's touched by a lot of people and that is conducive to harboring that virus long enough so that some other human can then pick it up. Okay. So think of it like, burrs, right? The plant that has a burr, it's just waiting, and so, but it can't move, so it's waiting for some animal with hair that it can then stick to, and then that animal transports it somewhere, and eventually it falls off, and then now it's moved on, right? So the animal is kind of like the delivery system for it, and so fomites will be things like water faucets, right? Or like I said, doorknobs are great ones. The dry erasers, or the dry eraser pens, like if I'm sick and I do this, and then I use this pen and I do a lecture and I write on the board and then I put it back in the tray, and then the next instructor comes up and they do this, and then they're like witching their nose. That's a great way to pass it along. So fomites are surfaces that allow the spread of not just viruses, any kind of microbe. And remember, these are non-enveloped now that we're talking about, right? So do non-enveloped viruses or do enveloped viruses last longer outside the host?
Non-enveloped last longer.
Non-enveloped, yes. So these kinds of things, like the shower, let's say you go into a public restroom and you use the shower there and you don't have flip-flops. There's all kinds of nice little papilloma virus particles probably in that shower just waiting to give you a wart on your foot. Okay. They usually regress over time. They can be removed physically or chemically. They can be burned off or frozen off, but the warts can recur if you have a good enough infection established. It may not just be one. There are the common seed warts. These are just painless and they grow usually on your hands, on the outside, not the palms, typically on the outside of your hands. Plantar warts are going to be more of an issue. They're going to be these deep, almost they have like these roots. When you remove it, it has these structures that look like roots going up in there. So it's very painful. You walk on it. And then genital warts, right? Common STD. All of these are from papilloma viruses. Okay. Papilloma also can also cause cancer. All right. So there are 9 different types of papilloma that are associated with reproductive cancer, and 2 of them for metastatic tumors. So metastatic means that the tumor is now spread to other places in the body. So think Henrietta Lacks, okay, and HeLa cells. Those were cervical cancer cells. And so the way to detect a cervical cancer or some other kind of cancer that's being driven by a papilloma virus is to do routine screens, one of which is the pap smear. And what that does is it allows the pathologist to look down here at these cells, and here's a normal cervical cell, and here's what's called a coilocyte. Okay, so these viruses have infected this cell, and then they have to exit, so they leave and they go into another cell, and it causes these cells in these nearby areas to clump together into this abnormal morphology. Okay, and you can also see that the nucleus is enlarged compared to this, and the cell overall is smaller, okay? So it just screws up these cells, and that can be detected by doing a pap smear. And the idea is that hopefully you catch it before it becomes metastatic or before it becomes a full-blown tumor.
My doctor told me, I asked her about like women that get it, and she just said that it's a really slow growing cancer and that most of the time it's because they weren't getting checked. And she said it could take up to 10 years. Is that true? Your wife's an OBGYN, right? So she probably very familiar with this, I'm sure.
Yeah, it is for the most part slow growing. Now, cervical cancer can come on very quickly, like we learned with Henrietta Lacks. I mean, from the time of her diagnosis to her demise, it was less than a year, and then she'd had symptoms for maybe a year before. So depending on the aggressiveness of it, it can be rapid onset, but typically, yes, it's from a lack of routine pap smear, I believe. And now there are vaccines, Gardasil for one, that has been strongly encouraged in teenagers to prevent this, right? So that it's not passed along. Papillomavirus isn't particularly, it doesn't impact males nearly as much as it does females. So, but they can be a carrier and they can pass it on to females. So, yeah, typically I think it takes a while. You don't get many, young females with full-blown cervical cancer due to papillomavirus. It takes a while. Any other questions on this? No? Okay. Thank you. And then parvovirus. These are definitely viruses that you might hear about if you get a puppy or if you get a dog in general. They need it, like it can pass through animals, like our pets. Cats can get it as well. It can be very bad for puppies because it can infect their heart and lead to heart disease and essentially, cardiac arrest. Not very common in humans. And this is an example of a DNA virus that is single-stranded. So parvo is a single-stranded, non-enveloped DNA virus. Okay. We made it. Okay. Before we get into RNA viruses, are there any questions about DNA viruses? I know we kind of blew through that, but anything you guys, or questions or comments on that?
What chapter was that we just finished?
24, I believe.
Thank you.
So the viruses are going to be chapter 6, chapter 24, and then chapter 25, which we'll go into now. Okay.
I have a question
Mm-hmm.
or like a comment.
Sure.
I actually got my adenoids removed.
Mm-hmm.
So what does that do? Because I was really small and I don't
Yeah.
know why.
Well, so when young people, usually when they get persistent ear infections, I don't know if you remember or if you were told that you had a lot of ear infections, but oftentimes that will be a reason for removing the adenoids. Also, if you get a lot of recurrent strep infection, they remove the adenoids because that is a site for infection. And so if they're gone, then that releases the burden of getting recurrent infections in the ear and then the throat.
Okay, thank you.
It doesn't mean that you're immune to adenoviruses because, as I said, it can infect the respiratory system and stuff like that. But yeah, I would imagine that's probably why you had them removed, because you just had recurrent infections.
Thank you.
Okay, so lots of interesting RNA viruses, I think. Now we're talking about things like influenza. We're talking about retroviruses, so HIV. We're talking about dengue fever, SARS, right? Polio, some of the hepatitis viruses, the diarrhea virus, rotavirus, measles, mumps, rabies, Ebola, really the ones that terrify us more so than not that the DNA viruses aren't dangerous and lethal and should be approached soberly. But the RNA viruses are the ones that that usually seem to send chills, literally and figuratively, in our bodies. And so one of the reasons is that RNA viruses have high mutation rates because they don't have the machinery to correct those mutations. So a DNA virus doesn't have nearly as many mutations. It doesn't mean that they don't mutate. Like even our cells can mutate, but when you're transcribing DNA, there are things there. The polymerase that is used for that has a self-proofing, like a proofreading mechanism on it that detects when there's the wrong nucleotide put in to that nascent RNA strand, and it will then excise it out and correct it. RNA viruses don't have that. And so as a result, they can mutate with high frequency. And so you get all these different varieties. And then also, you get things like influenza, which you get it once, you're immune to that particular strain, but then it swaps around some segments and it mutates, and to the point that your immune response no longer recognizes it. And so hence the need for a flu shot every year, which now we're coming into flu season. So if you haven't got your flu shot yet, I recommend it. Even if you're of the mindset that, well, how do I even know it's going to work? It's just a crapshoot, right? They just predict whatever they think is going to be the strain, and they make a bunch of vaccinations for that. Even if it's not the right vaccination for the right strand, a flu shot can boost your immune system and prime it so that if you do get the flu, the symptoms aren't as bad. Okay, so that's my little plug for the flu shot and why you should get it. Not to mention, it's bad enough to get COVID-19, but you don't want COVID-19 and a co-infection of the flu, right? So food for thought. Tables, don't need to memorize them, but you just there if you want to look and see how these RNA viruses pan out in terms of their tissue tropism. They're all over the place, right? So they can be in the skin, in the muscle, in the nervous system, in the GI tract, in the respiratory tract, the urogenital tract. All of these places have RNA viruses that have a specific tissue tropism. Okay. Let's talk first about the enveloped, segmented, single-strand RNA viruses. Okay, so what does that mean? Envelope means that it has an envelope, obviously. Segmented means that it has more than one segment of ssRNA or single-stranded RNA. Right? And so these are going to be the orthomyxoviruses, for example, influenza, Bunya virus, arenavirus, and so we'll talk about those a little bit more now. Influenza infects the respiratory tract. You have segments of RNA that enter then the nucleus. So this is the opposite example of an RNA virus that can then enter the nucleus, whereas most RNA viruses stay out of the nucleus. And once it's made its new viral particles, its little babies, they bud off the cell and they take the envelope, a little bit of that host membrane with them as the envelope. This is kind of the process of influenza. So at first, we have the hemagglutinin spike and the neuraminidase spike, and that is what gives each strain of the flu its name. H for hemagglutinin, N for neuraminidase. So H1N1 has a particular type of hemagglutinin spike, the first one, and neuraminidase, And if it's N1, it's going to have that particular type. They bind to the host cell receptors and then enter. Okay. And once they enter, they have an unfolding, right? It opens up and it releases all of these different segments. Okay. These segments of single strand RNA are going to go into the nucleus. And then from there, it's going to transcribe that into RNA, positive sense RNA, which, remember, if it's positive sense RNA, it can go straight to translation, right? If it's negative sense RNA, it's got to first make positive sense RNA, and then that gets translated, right? Okay. So these are negative sense RNA viruses that then have the positive sense that then leaves the nucleus and then goes back to the cytoplasm and uses the ribozymes and the ribosomes to then make proteins that the spikes, right, the hemagglutinin and the neuraminidase spikes, it's translating those proteins out, and then it's shooting those proteins into the cell membrane of the host. It replicates more negative sense RNA segments that also leave the nucleus and then go to the site at the membrane of the cell where those spikes have been intercalated in, and then it wraps it all up into a nice little particle and buds off. Okay, and that's the flu. That's what it does. Coronavirus is not like the flu. Coronaviruses do not replicate like this, despite what some people like to say. Glycoproteins, right? We mentioned that glycoproteins are just sugar and protein. Glyco, sugar, protein, obvious. And so these are going to be the hemagglutinin and the neuraminidase. There are 15 types of hemagglutinin. There are 9 types of neuraminidase. And depending on the strain, depending on how these segments swap from one particle to the next, if you have co-infection of 2 different influenza strains, they both go into that host cell, they both release their different segments of their RNA, and then they start intermingling, and you start packaging up some of the RNA segments from influenza type 1 and some of the RNA segments from influenza type 2, for instance, and then now you have a new combination that may be more severe in its infection, or may now be able to infect an animal, aka us, that didn't previously. Okay. Does that make sense? That's how the flu is able to basically come back every year, because it swaps around those segments and it mutates. Okay. Any questions on that? Okay. Now, if you look here, it says binding sites used to anchor virus to host cell receptors have a low mutation rate, but the site for antibody binding has a high mutation rate. Okay. So while the receptors have a very high fidelity to, well, the viral receptors have a very high fidelity to the host membrane receptors, the things that they can bind to, the part of them that gets recognized by the antibody that we've produced to previous infections, that site has a high rate of mutation to the point that antibodies may not recognize it. Bummer, right? There are 2 different types of drift and shift when we're talking about the antigens. Constant mutation is called antigenic drift. And so it's a gradual change of the amino acid composition. So the things that make the proteins that are then recognized as antigen. Or there's antigen shift, where one of the genes, one of the RNA strands, is substituted with a strand from another influenza virus, what I was talking about in that previous slide. Okay, so you have the human influenza virus, and it's undergoing some antigenic drift on its own, right? Slowly mutating. Then you have the duck influenza virus, that's doing some antigenic drift slowly on its own. But for whatever reason, those hemagglutinin and those neuraminidase spikes are both able to bind to the pig. And now we have a swapping, we have an antigenic shift where the segments of the human flu and the segments of the duck flu package into one. And that makes it a really nasty flu for us because we have not encountered that particular type. Okay. We don't have any exposure to the duck antigens because the duck flu isn't capable of infecting us. It's only when it mixes in another host that it then it gets enough of the human segments to allow it to bind to our human cells. Okay, hopefully I didn't belabor that too much. Hopefully that makes sense. If you get the flu, Tamiflu is probably one of your best bets. It comes from Genentech. I used to work there, so that's kind of a shameless plug for Tamiflu. But you got to take it. You got to take it kind of early before you get the full-blown infection. And then obviously the annual vaccine is recommended. Okay. Any questions on flu? Nope. Okay. Bunya viruses and arenaviruses. Very, very nasty. Okay. Biosafety level 4. What does that mean? Well, biosafety level 1 means there's really very little precautions that need to be taken. Biosafety level 2 means, yeah, you should probably have a hood at least to work under. Biosafety level 3 is really getting into much more PPE wearing. Biosafety level 4 is where you need to start wearing the gowns and going into a showering station and things like that. These are bad. Bunya viruses are vector-borne, so ticks, other arthropods can spread this, and it can lead to hemorrhagic fever. So you're bleeding and you have a fever. It's nasty, right? Hantavirus, sin nombre virus. These are things that have emerged relatively, recently, but the hantavirus is terrible. Okay, it's got a 33% mortality rate. So that, I don't know if that seems high to you or not, but 33% is pretty high, right? 33 out of 100 people that get it are going to die. Can be carried by deer, mice, even airborne if the waste. So remember, all these viruses get shed out through our waste, and then if you have dried fecal material from these animals that then gets aerosolized as it decomposes, now you have those viral particles, and so it can infect you by breathing it in. And the deer and the mice, they get it from these arthropods that bite them or live on them. Arenavirus, again, loss of fever, hemorrhagic fevers, often associated with some kind of rodent infestation. So it comes from the rodents, and then you get it because the rodents are living in your house and they're pooping in your house. And then if they have these viruses, they're shed out that way. And, maybe they're in your pantry, near your food. So then, it gets in your food. Maybe, again, like I said, it gets aerosolized and you just breathe it in, but it's pretty gnarly, okay? So that's the segmented RNA viruses. How about the non-segmented RNA viruses? These are going to be paramyxovirus. So parainfluenza, mumps, measles, the rhabdoviruses, so like rabies, okay? Coronaviruses, toga virus, so your rubella, your MMR, right? That's going to be your mumps, your measles, and your rubella. Flaviviruses, think hepatitis C. Flaviviruses can also be not just hepatitis C. They can be really bad or other RNA viruses as well. And then filoviruses like the Ebola virus. These are non-segmented. Now, am I going to make you guys memorize every single virus that I've put up here and whether or not it's a double-strand DNA virus or an enveloped DNA or a non-segmented? No. What do I expect you to know? I expect you to know examples of all of these types, okay? So it might be something like, what is an example of an enveloped non-segmented single-strand RNA virus? And you should know one of those, okay? I don't know, maybe two, but point is you don't need to memorize all of these viruses that we're talking about. Paramyxovirus, parainfluenza. So the interesting thing about this is that it can form this syncytium that I mentioned on Monday, or these multinucleated giant cells, because what happens is one infected cell is touching another non-infected cell, and the new virion just buds out of that cell into the next cell. And then it does its thing, right? And so then you have all these cells kind of stuck together and they're all now infected. And so you get this syncytium as a result. Okay. Do you need to know that paramyxovirus and pneumovirus, like the respiratory syncytia, do you need to know that that's particularly involved in multinucleated giant cells? No. Do you need to know what a syncytium is? Probably. Okay. Mumps, most of them, not most, I guess 40% are subclinical. So we don't really even know that we have mumps in 40% of the population. This is an example of a virus that's only passed on from human to human. You can't get mumps from your pet or the mice that live in your house or the deer or some kind of insect that bites you. Okay, it's just from being around humans. And you get this swelling. And again, it's not super common. I guess it's becoming more common now. But since the use of the MMR vaccine, most people, you know, that are adults now haven't had to deal with this. Measles, very contagious, right? We're seeing this now start to reemerge again as vaccination rates, MMR vaccinations are going down. I believe it was the MMR vaccine that was inaccurately described in the Andrew Wakefield paper, in the Lancet paper, that claimed that there was a correlation between vaccination and autism. The paper has since been retracted because it was shown that the data was manipulated. But that data, that manipulated data, I believe was showing a link between the MMR. So the vaccine that treats measles, mumps, and rubella. Rabies, it's a rhabdovirus. Remember, rhabdo is like a tube or a rod. And so it's right here. What kind of capsid does it have? Based on this formation, what would you think?
A helical.
Helical, yes. So see here, the capsid here, it's helical. right? It's going, it's spinning around to form this tube, okay? And rabies, as we know, comes from animals. I think, right? Old Yeller had to be put down because of rabies. If you guys like The Office, Michael Scott wants to have some kind of race to bring awareness to rabies, which, you know, what he didn't realize is that there's actually a vaccine for rabies, and then it's not a very common thing anymore. So luckily, we don't have to deal with that. You can see here, though, that it's still widely distributed in the US in our animal reservoirs. So primarily in our neck of the woods, it's going to be skunks, as well as a large swath of the Midwest. But in the South, we're talking foxes, and the East Coast, mostly raccoons. And so what happens is our pet, if it hasn't been vaccinated, will get in a skirmish with one of these critters, and we'll get bitten, and then we'll get rabies that way. Coronaviruses. Oh man, did you think that you had learned everything you needed to know or wanted to know about coronaviruses? Well, in case you aren't sick of coronavirus yet, let's go into the biology of them a little bit here, okay? It's a relatively large RNA virus. It has these distinctively spaced spikes on the envelope, okay? Which gives it that corona halo-like appearance resembling the sun, hence corona, right? Common in domesticated animals, or in the case of SARS-CoV-2 and the initial SARS, most likely animals that are being traded and being in proximity with each other in these wet markets where they're in these cages stacked on top of each other. And if they have coronaviruses, they can recombine into a particular host and then become infectious in other hosts like humans that weren't previously capable of infecting. Okay. Everybody, coronaviruses are pretty prevalent. Very few of them are like the one we're experiencing now. Most of them are just the common cold. But they infect a wide variety of mammals. And bats are the biggest reservoir for coronaviruses. And what happens is the bat, because they can fly, have a broad range of travel, and they defecate as they're flying, and the coronavirus is shed in those, in that poop, and then it falls. Let's say you have a bat that likes fruit, right? It goes and it poops in the orchard, and then oftentimes they'll let their livestock kind of graze on the fallen fruit. And then that livestock has eaten that apple that fell out of the tree that also was pooped on by the bat. And then that's how these viruses recombine into something that now can infect a human. Okay. We're not exactly sure on this new SARS-CoV-2 virus, how it mutated, but we think it came from a bat that then jumped into a pangolin. And if you don't know what a pangolin is, it's a very cute little scaled mammal that is a hot commodity on the illegal trade. And so, so we think that it might have come about that way. Okay. Airborne transmission, 9% of cases fatal. That is, that is the typical common cold flavor of coronavirus. Of course, with these, with SARS-CoV-2, that's different now, right? But 9% is high still, right? I mean, 9% is that's 9 out of 100 people, right? When you put it in that perspective, it's not a small amount. Rubella, single strand RNA, has a loose envelope. So it's not tethered down as strongly with these matrix proteins. And oftentimes in young people, they're the ones that get rubella. And again, we have the MMR, so it shouldn't be as common as it once was, but it's probably somewhat on the incline now. Hepatitis C, caused by flaviviruses. These are acquired through blood transmission. So if you get a blood transfusion that wasn't screened properly, or you get a dirty needle, you can have transmission of Hep C that way. And we already talked about Hep C, so I won't belabor it. But and actually, I need to update this. So clearly, this slide, clearly this slide was from last year when there was no vaccine. But that right a line through that. That's no longer the case. Okay, arboviruses, arthropod vectors, mosquitoes, ticks, flies, that kind of thing. Lots of different viruses in this family. And some of them are flaviviruses, some of them are bunyaviruses. So nasty, nasty viruses that are lethal. That said, most of them are somewhat benign. Some may cause severe encephalitis. So as I mentioned, the Eastern equine encephalitis virus had a recent outbreak in the North Midwest. And some people will die from that. Not a lot. It doesn't spread a whole lot, thankfully. But some can be life-threatening and cause hemorrhagic fever. Some of these flaviviruses in particular, and bunyaviruses. When you look at the distribution, it's mostly in the tropical region. So around the equator, where insects don't get killed off when the cold comes. And so again, as we start thinking about the average global temperatures rising, what kind of ramifications does that have on things like these arboviruses that now will start migrating more and more north and more south in regions that usually had insects die off significantly. And now, we see the spread of a virus that was more contained again in the tropical regions. West Nile virus, that's an arbovirus. As I mentioned, the Eastern equine encephalitis virus, chikungunya, that's a pretty bad one. Headache, joint pain, nausea, maybe, occasional death from that, but it's very uncomfortable. It's a really, really debilitating viral infection. And so it has it harms the economy because it's in these areas where people, they don't get paid if they don't work, right? They don't have sick leave. And this can hold them up for a while they're experiencing these symptoms. And so every day that they're not working is a day they're not making money. And the money they are making isn't that much anyway. So has pretty big ramifications on the economy in these tropical regions. And the Zika virus, we kind of know about Zika virus a few years ago. It was on the rise in the tropical areas in Central America and in South America. And so the idea of traveling to those areas, especially if you were a mother who was pregnant or considering getting pregnant, because it was found in male semen as well. So even if you weren't pregnant at the time, but you were thinking about trying and you'd recently traveled to South America, that could have been an issue. And of course, the big issue with Zika is that it harms the child in development and causes microcephaly. Hemorrhagic fever, yellow fever, dengue fever. Okay, these are typically not in the US. These are going to be most likely in the tropical regions. That doesn't mean that they can't make their way up here at some point though. HIV, came out in the 80s. There's a documented case going back to the late 50s, so it's been around for at least then. We're not sure how it emerged. There are all kinds of hypotheses, but no one really has the definitive answer for that. And it infects your immune cells, right? So you don't usually die from the virus, you die from something else that your immune cells can no longer fight off. It's a retrovirus, and so what it does is that it integrates into host DNA. And I mentioned this a little bit on Wednesday, I think. Once that happens, you cannot get rid of it. It is in your DNA, right? So you have a lifelong association with this, with retroviruses. And they use something called reverse transcriptase that then allows them to take a double-stranded DNA segment and make it from the single-strand RNA genome. Okay. Now, any questions so far? We're almost done, but you know, with HIV, there was an interesting treatment last year, I think, where they essentially took these immune cells from a patient who had a mutation, a rare mutation that could not be infected by HIV. They took those immune cells from this donor, and then they used chemotherapy to destroy all the immune cells in a patient with HIV. And then they grew up these immune cells and they put them in that patient and effectively cured that patient of HIV. And that's really, I mean, that's the only way to cure somebody, because it is integrated into your genome. Unless you get rid of that genome, you don't get rid of the virus. Okay. Well, the problem is it was very, very harsh to be a donor. Like that person who they found several patients that were willing to donate their immune cells. And but I think one of them actually didn't survive the procedure. So it's probably not going to be an effective way to treat HIV. However, if you think about things like CRISPR, which we just found out, right? The two women who used CRISPR, which is a mechanism of bacterial cells to fight off viral infection. The women were awarded the Nobel Prize because they found out a way to use that and create a tool for genome editing. Perhaps we can make immune cells that have that mutation in vitro, right, in a dish, and then put that into the patient. I don't know. I think that'll be the wave of the future, essentially. This is, if you want to look at this, you can. This won't be test or quiz material. It's just showing you the epidemiology of HIV in the US, and where it's highest, where it's lowest. I'll let you look at that on your own. Lymphotropic viruses, think leukemia, right? They are acquired. They're not inherited. So a leukemia is a form of cancer, and cancer can come because you inherit a mutation, or from your parents or parent, or you acquire that mutation. Okay, and it's in your life, whether it's being exposed to some kind of carcinogen or whatnot. In this case, it's a viral infection. And various forms of survival. Some are acute, some are chronic, some are life-threatening. Most of them, I guess, are probably life-threatening, but some people have a much better prognosis than others. Okay, non-enveloped, non-segmented single-strand RNA viruses, such as the picornavirus, which we said pico is small RNA, so small RNA virus. Enterovirus, so polio, rhinovirus, so common cold virus, and cardiovirus, which is not very common, but is around. Polio, of course, is, we had the vaccine in the 50s from Jonas Salk, and that really, squashed polio. But before that, it was a terrible, terrible infection that paralyzed and killed countless people. And also, the polio virus vaccine was validated in HeLa cells. So thank you, HeLa cells, for one of the many benefits, that being the vaccine against polio. Mostly eradicated. Again, we're seeing this on the rise in some places due to anti-vaccination philosophies. I won't talk about that. That's just a little bit about the vaccination, prevention. Rhinovirus, 110 serotypes, common cold. Lots of them are circulating at a given time. So we're getting into cold season right now. Some of you will get a rhinovirus or rhinoviruses this fall. Some of your family members will get it. You'll get this common symptoms of a cold, right? Chills, fever, sore throat, runny nose. And there's no real vaccine. There's no way to treat the infection. You just treat the symptoms, right? You take some meds and to relieve those symptoms. Reovirus, again, it's an upper respiratory virus. It can also cause stomach irritation. Rotavirus is going to be a diarrhea virus from oral fecal contamination. These often are outbreaks in daycare facilities where kids, I don't know if you have kids or not, or if you've worked at a daycare, but there's that stuff is, feces is not literally everywhere, but it's all around, and kids just play with it and play with each other's. It's gross. So oftentimes you get an outbreak of rotavirus and a bunch of kids get sent home because they have diarrhea. And of course, if you're in a developing country and you have diarrhea, that could lead to dehydration and death. So it's serious depending on where your socioeconomic status and where you are. All right, I'm gonna stop there because that's perfect. 1:35 on the dot. There's a couple things here. Genetics, yeah, we won't get into that. That won't be test material. Just a few slides. Any questions? That's a lot of viruses. Wow. Again, virology, like immunology, can be a class in and of itself. I took a virology class in grad school, so we had to do it in 3 lectures. So a lot of information. Most of that is just for you to understand the variety. And when we talk about viruses that specifically infect us, give you an idea of what they're like, on the microscopic level. And so again, you don't need to memorize all of these slides. You just need to know examples of these different types. And that's going to be enveloped, non-enveloped, icosahedral, helical, DNA virus, RNA virus, double-stranded, single-stranded, that kind of bigger picture level. If I say, what is an example of an enveloped single-strand RNA virus, be prepared to provide something, okay? I'm not concerned with signs or symptoms per se. I don't think that that's as important because, again, most viruses have a similar type of manifestation, unless they have, cutaneous lesions or hemorrhagic. But any questions on that? That wraps up viruses. We're going to move into bacteria next week. So any virology questions? Okay. again, Monday, we will be working on our proposals. So be prepared to talk about your topic a little bit. And that'll be the assignment. My assignment for that lab will be for you to just submit the notes from the brainstorming session that you got, if you got some ideas as a result of talking with your peers. Okay. All right.
I do have a quick question. If you
Yeah.
know, going back to the one with HIV, the donor, you said one of the donating process was might have actually killed one of the donors. What was it? What did they have to do to get that?
Well, what they did was they found these people who had immune cells that had a mutation that prevented them from being infected by HIV. So they harvested the immune cells from that person. And I believe there were 2 or 3. And one of the donors, he or she, I don't know what their gender was, expired. And I think it's because they took their immune cells. And I don't know if it was, I don't think it was the actual extraction, but it was the consequence of having limited immune cells, you know, so that you maybe an opportunistic infection came about. I don't recall the exact details, but I remember thinking, well, wow, this is great. We have these superhumans that can then, you know, every once in a while donate their cells like we donate blood, and we can treat those that have it. But then one of the donors died, and then it became very apparent that that's probably not an effective therapy. You know, I mean, the approach
Well,
of getting them from a donor is not going to be feasible.
okay.
So that's why I say take immune cells and put them in a dish, and then use CRISPR to create that mutation. And you know what? Maybe now that I think, I'll have to look into this, Christine, because the more I think about it, maybe it was the person that got the transplant. Maybe they were the ones that expired, but I want to say it was the donor.
Okay.
But you know, it's a very harsh therapy, because you are, like I said, you're undergoing chemotherapy to essentially destroy all of your immune cells, and then you're repopulated with this immune cell that has a mutation. And so there's that period of time where you have no immune cells in your body, right? And so if you catch some nosocomial infection in the hospital while you're having this procedure done, it could be life-threatening, to say the least, right? So it's radical. It's a radical treatment, but it's the only thing we have right now.
Okay, yeah. Awesome. I had heard about that while back, but I didn't really know about some of those the results.
Yeah, it's pretty cool.
Yeah.
And that's why I say CRISPR, you know, you guys wait and see. You know, it'll take a while to get the unintended targets to square that away, so that when you are using CRISPR on someone, that you're not editing other things that you're not supposed to. But I mean, someday we will have CRISPR or some form of that editing to do these kinds of therapies, whether it's someone with cancer or someone with an integrated viral, you know, infection, or, you know, the world is the oyster at this point with CRISPR, but it's in its infancy right now. But someday I think it'll be a big thing. It's exciting. All right. Well, I won't keep you guys any longer unless there's other questions, but have a great weekend. Have a safe weekend, and be prepared to discuss your topic on Monday. I'm excited. We'll go into bacteria, so it should be fun.